الحمد لله دكتور الحمد لله.
First, we'll discuss not rickettsia. Rickettsia is the final one. Hemophilus. Hemophilus influenzae was actually first discovered in 1892 by Richard Pfeiffer. Richard Pfeiffer thought that this was the bacterial cause of influenza, which was actually incorrect, and influenza is actually caused by only a virus. But this type of bacteria is somehow associated mostly with influenza. Why? Because it requires the heme, which is actually produced by the pneumococcus or the staphylococcus too. And influenza actually also requires the heme in order to activate its surface antigen اللي هو hemagglutinin. So it was actually incorrectly named influenza, but since the name actually went on, the bacteria continued to be called Haemophilus influenzae because of its requirement to heme, and it was discovered in the influenza pandemic. Now, this bacteria is actually the most troubling cause of meningitis, especially in infants. First year of life, Haemophilus influenzae, especially type B, is responsible for 90% of death in infants because of meningitis. Now, thankfully, due to the discovery of the vaccine, the percentage actually decreased. Lately, this bacteria was showed an outbreak in Iraq, and the percentage was actually according to the other causes of meningitis, viral and bacterial, it was 2.75%. And this percentage was in the last 2019, while in 1988, 1989, the percentage was much more higher, 13.8% of the causes of meningitis, viral and bacterial. And this drop in percentage is actually related to the vaccine. This microorganism is actually a normal flora of the upper respiratory tract. It is small gram-negative pleomorphic bacteria, which requires enriched media and certain derivatives for its isolation. Now, the pathology of this bacteria actually depends on its capsule, mostly. The capsulated are the highly virulent, the non-capsulated are mostly the normal flora. The capsulated is actually the antiphagocytic antigen of this of the virulent factor that causes meningitis, epiglottitis, cellulitis, bacteremia, and if it disseminates, it might lead to septic arthritis and pneumonia. While the non-capsulated form of H.influenzae could induce otitis media, and it is up to till now, it is the second most common cause of otitis media. Sinusitis, conjunctivitis, it might exacerbate chronic bronchitis, pneumonia, and bacteremia in adults. But this actually, the non-capsulated is less virulent than the capsulated form. The biogroup Aegypticus is responsible for the pink eye or knocked weak conjunctivitis. Another strain of the Aegypticus is the Brazilian purpuric fever. Now, this one has got a high mortality rate in children, especially between the ages of 1 to 4 years of age, and it is responsible for purulent meningitis, bacteremia, and high fever. The sexual transmitted species of Haemophilus is the Duckery. This is a sexual transmitted, it is capsulated, it has a pili which helps it to attach, and it also produces toxins. It causes chancroid, a genital lesion that progresses and forms papules. I think I have a picture right here of a chancroid. It is mostly found on the external genitalia or even on the skin near the genitalia. It has a yellowish and a gray base with an inflamed red color margin. How do we differentiate it from the syphilitic chancre? This type of chancre is actually painful, especially in men, but it is less notable in females. So this is mostly caused by Duckery, a sexual transmitted form of the bacteria. Haemophilus parainfluenzae is actually an opportunistic pathogen. It has less virulent than the H.influnzae, and it is part of the HAC group, the bacteria associated with prostatic valve endocarditis, and it might also induce pneumonia, but it's actually subacute. There's a low virulent microorganism which is part of the bacterial vaginosis. Now, this Haemophilus vaginalis, it is not sexually transmitted, but it is associated with another 25 microorganisms which actually causes bacterial vaginosis. And one of them is the Gardnerella vaginalis. Grouping these microorganisms will cause a yellowish purulent discharge from the vagina in females, especially in the reproductive stages. Married females are mostly fertile, are mostly exposed to this type of infection. Now let's speak more deeply in the H.influenzae. Why it is important? Because it is responsible for 90% of mortality in infants, meningitis. Now, since the vaccination, the percentage is actually dropped. The encapsulated H.influenzae contains a capsule polysaccharide. It is 6 types from A to F. The B type is actually composed of a polyribose phosphate capsular protein. The somatic antigen consists of the outer protein, and it has an lipooligosaccharide endotoxin, like any other gram-negative bacteria. The HAC group right here, I gave you the causative. It is the Acinetobacter, Aggregatobacter, Cardiobacter, Echinella, and Kingnella. All these are responsible for prostatic valve endocarditis. That means after surgery replacing the heart valve, patients are exposed to this group of bacteria causing endocarditis. Gram stain of this microorganism is very unique. It is small, short, pleomorphic gram negative bacilli, or mostly cocobacilli. The short chains. In culture, the morphology depends both on the length of incubation on the medium, and 6 to 8 hours rich medium shows a small cocobacilli like this. But if it is left for more than that, it will show this long form of pleomorphic arranged bacilli, like this. So this is H. influenzae in an acute infection. This is after 24-hour incubation on a selected media. The selected media on chocolate agar, it is flat, grayish, translucent colony with a diameter of 1 to 2 millimeters, presented mostly after 24 hours of incubation. It actually doesn't grow on sheep blood agar. Why? Because sheep blood agar could not supply the requirements of the growth of this bacteria, which is the V factor. The V factor will be taken if we culture with it Staphylococcus aureus, causing this satellite phenomena. You can see this is the colonies of Staphylococcus, and since staph produces the nicotinamide adenine dinucleotide factor, which is factor V, the bacteria will grow like satellites around the colonies of the Staphylococcus aureus. On chocolate agar, they are translucent, non-hemolytic type of colonies, small. So as a major, this bacteria will require two factors. The first is the X factor, which actually supplies the heme. And the second is the V factor, replaced by nicotinamide adenine dinucleotide, or sometimes even other coenzymes. Now, let's discuss the pathophysiology. إلى حد الآن عنده سؤال على هاي البكتيريا؟ ولدي؟ تفضل. النيسيريا طبعا الكولونيز مالتها تختلف، ميوكويد زين وتكون ديبلوكوكاي وجرام نيجاتيف. هذه البكتيريا تكون كوكوباسيلاي جرام نيجاتيف. واذا تشوف بالسلايد شلون تطلع عندنا، هاي رقم واحد. رقم اثنين اللي هو كلش مهم، هاي البكتيريا ما تنمو على الشوكليت اجار بشكل طبيعي. احنا غالبيه الشوكليت اجار هو شيب بلاد اجار، فاللي ينمو عندي النيسيريا. انا عم تنمو هاي البكتيريا لازم الميديا بها الاكس والفي فاكتور، فاكو ريكوايرمنتس خاصه على مود يصير الجروث لهاي البكتيريا. النيسيريا نادرا ما تصير بالانفنتس قبل ما يصير عندك conjunctivitis, severe conjunctivitis, mucopurulent conjunctivitis، العين تورم. هاي اذا انتقل النيسيريا meningitis from the or the gonorrhea from the mother to the child. اي ف rarely it happens in infants. الانفنتس هي mostly H.influnza the cause. فاحنا عم ننمي هاي، لازم عندنا هاي الساتلايت فينومينا يلا تبين عندنا. بعد احد عنده سؤال دللني. اي يعني اي او الشوكليت اجار نستخدم به الهيم، نستخدم به هورس بلاد، ما نستخدم شيب بلاد. اي على مود ينمو عندنا. طبعا اكو لها هي ميديات خاصه، بس الهورس بلاد بي ذا تو فاكتورز، الاكس والبي فاكتور. اوكي. بعد احد سؤال عنده؟ اوكي الو زين يلا. For this pathogen or this microorganism, in order to induce the disease, it requires a large infective dose. يعني هاي فالنقطة كلش مهمة، مو من ليش؟ لأنه هي احنا من البداية قلت لكم اكو نون كابسوليتد موجودة بالنورمال فلورا of the upper respiratory tract، وعم تصير كابسوليتد هي بس مجرد بلازميد بحيث تتحول الى كابسوليتد. فلازم يكون عندي اني فد فاكتور يقلل المناعة بحيث يسمح لهاي البكتيريا انه تنمو. فدائما نلقاها associated with viral infection، ولهذا هم خربطوا بيها وعبالهم هي البكتيريال cause of influenza، لانه يعني it is highly associated with influenza as a virus. فلازم عندي large dose ولازم عندي viral infection to start the infection. الcolonization to the mucous membrane، وبعدين تبدي تدخل blood stream. The presence of antibodies complement phagocytes determines the clearance of the bacteria. الفاكسين انقذنا من هاي البكتيريا. العفو. مجرد اخذنا السيرفس انتيجين مالتها وسوينا ربطناه بكارير وطيناه للجسم بحيث البي سيلز بدات تنتج انتي بوديز ضدها، وتحولت الى IgA secretory antibodies. صار colonization مال هاي البكتيريا بالupper respiratory tract impossible. فحتى الطفل اللي مثلا يعني انصاب بهاي البكتيريا he is liable for reinfection unless antibodies develop. إذا the first days of his life he's immune. Why? Because the placenta will transfer the antibodies to the child. But after that, the child is more prone, or sorry, the infant is more prone for the infection by this microorganism. طبعا الميجر فيرنت فاكتور هو الانتيفوغوسيتيك nature of the influenza type B capsule. هي هذه virulent factor. The absence of the capsule makes it non-virulent. Major defenses against this microorganism is the alternative classical pathway والantibodies. طبعا هنا الانتيبوديز والالترناتيف والكلاسيكال باثواي حيشتغلون كأوبسونينز. It will attract neutrophils, macrophages to the microorganism to destroy them. وبنفس الوقت حيسوي لي الكومبلمنت كومبلكس on the surface اللي يسوي لي damage للمايكروورجانيزم. فالانتيبودي والalternative classical pathway it's actually the major component in our defenses against this microorganism. Antibodies against the group B H.influnzae plays a primary role in confirming immunity. When newborns are more likely to be infected, therefore there are a high risk of repeated infections إذا ما صار عند الطفل immunity. And actually the action of antibodies in even non-vaccinated children will develop at the age of 5 years. يعني حتى الإصابة مالته بطيئة وما أدت إلى meningitis بال5 سنين تتكون immunity against this microorganism. هنا هاي نقطة مهمة. The conjugated vaccine does not produce immunity against the non-typable H.influenza، يعني اللي ما عندها شو اسمه التايب B capsule. And this actually caused the increase in the infections by this type of microorganisms. طبعا it is mostly responsible for otitis media. It might cause chronic bronchitis, sinusitis, and conjunctivitis. With the pneumococcus, they are the high suspected microorganisms to cause otitis media. طبعا الهيموفيلus ducrei is responsible for sexual transmitted it's part of the sexual transmitted diseases. It causes an ulcer or an open sore. شوفوا هاي ulcers of Ducrey, yellow grayish base, painful in men, less noticeable in females. وهذا هي البروبلم because the transmission will become more through the female side. اللاب diagnosis طبعا هنا احنا عندنا sample collection اللي هي الباس CSF, chancroid ulcer, spinal fluid. All these are sources for the microorganism. Direct detection هو بالdirect agglutination test, latex, which is actually مو كلش more sensitive than the gram stain. الجرام ستين of Ducrey فشي significant اللي يصير على شكل school of fish. شايفين بعالم البحار التقارير من تطلع الأسماك شلون تمشي بمجاميع. Same thing for Ducrey or sometimes even a railroad track. The arrangement of the microorganism فشي significantly واضح نشوفه. real time PCR طبعا هسه المالتيبلكس بي سي ار انقذنا يعني بس ندخل بي سمول سامبل مال سي اس اف وبكل الاورجانيزمز الفيرال البكتيريال الفانجل which could induce meningitis والمالتيبلكس بي سي ار يصير لها ديتكشن خلال ساعه. بس المشكله بالمالتيبلكس بي سي ار يريد لود يعني مو شو اسمه واحدة اثنين مايكرو اورجانيزم حسب التشخيص، لا يريد له كمية شوية كبيرة من البكتيريا يلا يعطيني بوزيتيف ريزلت. ولهذا دائما مالتيبلكس بي سي ار نحاول نسوي اذا بدنا نسوي كونفرميشن للمايكرو اورجانيزم افتر كلتشر. طبعا سبيسيمنت من ما قلنا يراد للفي والاكس فاكتور. فذا بيست جروث ميديا از ايزوفيتال اكس enriched chocolate agar. وطبعا هي requires 5% CO2 اللي هي fastigious microorganism و also could grow at 35 to 37 centigrades. طبعا the characteristic on the blood agar and on the chocolate is that they lack hemolysis on blood agar. ما تعطيني zone of hemolysis. هي translucent مثل ما قلنا small colonies. طبعا ورا ما يصير growth عم نميز ال H.influenzae عن باقي non-typable microorganisms or other microorganisms. نقدر نسوي هذا التست هو ال XV تيست. نجيب الشوكولات agar او blood agar ونخلي عليه two strips, one the X factor and the other the V factor. Certain microorganisms will develop against the X factor, others to the V factor, but H.influenzae will develop in between them because they require both of these factors to grow. As I stated, it is responsible for 90% mortality of meningitis. It is highly susceptible to penicillin. بس المشكلة هسه بعد يصير عندنا development of resistant 25% produce beta lactamase. And all strains are susceptible to third generation cephalosporin, carbapenem, and cefotaxime. Intravenously is طبعا هو يعتبر therapeutic. الاميونايزيشن طبعا هنا ننطي سكيمد فاكسين اللي هو غالبيته هو الانتيجين بس ويا كارير at 2 4 6 months. بعدين ننطيهم بوستر at 2 and 4 months apart. العفو بوستر ننطيه بال 12 و 18 months. طبعا هذا يكون ويا combination ويا cranibacterium and Neisseria toxin نسميه DTAP vaccine. ورغم من هذا هم صار عندنا اوت بريكس، بس الكيسات تكون مايلدر. والاوت بريك اللي صار عندنا هو يعني mixed between them both الانفلونزي and parainfluenza. اوكي. البكتيريا الثانيه اللي حنحكي عليها هي البورديتيلا بيرتوسيس ووبين كاف. فد قحه مميزه للوبين كاف لازم تعرفوها ولازم تسمعوها. اني خالها بالكلينيكال فيتشرز هنا. خليني اسمع لكم اياها قبل ما نحكيها. تسمعوها؟ اوكي، دقيقة. ليش ماكو صوت؟ تسمعوها؟
لا دكتور ماكو اي صوت.
دقيقة، مش لازم. دقيقة.
Whooping cough. It's a bacterial infection that's risen to epidemic levels in some parts of the U.S. In adults, the infection is spread to a baby who hasn't yet received a full course of vaccinations. Whooping cough can be very serious. Here's more on how to handle this disease.
Are you being shy?
21-month-old Pedro Escueda's symptoms started out like those of a typical cold.
He had a little runny nose and started having a little cough.
But over the next 4 to 5 days, Pedro's mom, Chelsea, says the cough got much worse.
It was nothing I've ever heard before. He was coughing and doing the whoop. He'd vomit.
And he couldn't.
بس ملاحظة،
whooping.
الووب ده تسمعوني مو ولدي؟
نعم دكتور.
ووب هو الصوت الطفل ياخذه بالانسبيريشن. ليش؟ هو هنا عندي انا ميوكو بيورلنت بلادي سبيوتم دي يصير بلاج للتيوبز، فالانسبيريشن ورا القحه القويه يصير بيها ساوند. فاذا تنتبهون على الووب هو مو بالقحه، هو من ياخذ الانسبيريشن ورا القحه. فشوفوها هسه.
It's a bacterial infection that's risen to epidemic levels in some parts of the U.S. In adults, the symptoms can be mild, but if the infection is spread to a baby who hasn't yet received a full course of vaccinations, whooping cough can be very serious. Here's more on how to handle this disease.
Are you being shy?
21-month-old Pedro Escueda's symptoms started out like those of a typical cold.
He had a little runny nose and started having a little cough.
But over the next 4 to 5 days, Pedro's mom, Chelsea, says the cough got much worse.
It was nothing I've ever heard before. He was coughing and doing the whoop. He'd vomit.
And he couldn't catch his breath. When they took Pedro to the doctor, the coughing had subsided, so they figured maybe it was just a cold or some other infection. But Chelsea knew there was something else wrong. So the next time Pedro had a coughing fit, her husband got it on video. A nurse practitioner saw it and diagnosed it right away.
And she saw it, she goes, that's pertussis.
Whooping cough is also known as pertussis. It's caused by a bacterial infection that is spread from one person with whooping cough to another person.
What is he doing?
Dr. Thomas Boyce says whooping cough is particularly tough on kids younger than Pedro, who haven't had the full course of vaccinations given at 2, 4, and 6 months.
They're particularly susceptible to getting infection, and because of their small airway, they're particularly susceptible to having very severe disease.
You see, the infection happens in the airway or trachea. The pertussis bacteria produce toxins that irritate and inflame the airway. This causes the severe, repeated cough that lasts for months.
They call it the 100-day cough.
Pedro and his entire family took antibiotics to treat the infection and prevent further spread. Now he's back to health after months of illness. Dr. Boyce says babies usually get whooping cough from adults who have a mild infection. He says that's why it's important for adults to get booster vaccinations every 10 years. Ask your doctor for the tetanus diphtheria shot that also includes a pertussis booster. For Medical Edge, I'm Vivian Williams.
اوكي ولدي، هو السؤال ديكي هو الوhooping cough او ال 100 day cough or the احنا مثل ما احنا نسميه السؤال ديكي. It is actually not a new disease. In the 16th century, Goldman actually reported a total of 48.5 million cases annually. The problem with this organism is that there's no vector and there's no host but humans. So it is transmitted from one human to the other. And it wasn't until 1906 when Jules Bordet and Octave Gangio produced the culture for this microorganism and were able to diagnose it and document it as Bordet ganglion bacilli. And it was further called Bordetella. Now, still it is a common problem in our country. There was an outbreak in 1996 in Basra, where 41% of the cases isolated were actually from the Bordetella pertussis. And there were some mixed Bordetella pertussis with Bordetella parapertussis. The problem is that these infants, they got the infection from their mothers. And since that time, the program of vaccination was actually enforced. And the cases today is actually approximately 80 cases per year. Still fluctuating because Iraqi families, they don't take the program as a serious matter. They think that since their child is healthy, it is better not to vaccinate him. And still we face a, let's say, critical problem in vaccination programs is that all our society fears what we call vaccine. They think that we will inject them with a toxin or a poison, or that these vaccines are not well handled or stored. فصراحة يعني احنا واجهنا صعوبة حتى بالكوفيد فاكسين. يعني الكوفيد فاكسين مش قد حكيت عليه ومش قد نصحت العالم عليه، لكن مع الأسف هواية حتى من زملائي إلى حد الآن أكو أطباء الغريب أنه ما مقتنعين باللقاح. فالفكرة من الفاكسين أنتوا لاحظوا الدروب اللي صار عندنا بالفاكسينيشن، وهاي البكتيريا يعني من البكتيريا نقدر احنا نقضي عليها بس مجرد انه ننطي فاكسينيشن. والمشكله بهاي البكتيريا انه it has got complications. It could lead to meningitis. The program of vaccination is actually important and requires enforcement. وان شاء الله enforcement تيجي من عندكم بالمستقبل ان شاء الله ولدي. So let's carry on. There are three major species that could cause infection in human beings. طبعا اللي هو بالحقيقة two major species, Bordetella pertussis with parapertussis. Bronchoseptica is actually an animal microorganism. طبعا هو why اكو من عندها البوردتيلا many microorganisms, many species, but those are actually the major two pathogens for strictly human being, pertussis with parapertussis. Pertussis causes pertussis, and the parapertussis induces pertussis, but to a less severe degree. The bronchosepticum, we are detecting it in immune-compromised patients, inducing pneumonia. Holmesi, it actually produces a pertussis-like illness, cough, chronic cough. طبعا pertussis is highly contagious, especially in the catarrhal period. يعني the pertussis passes into 3 stages: catarrhal, paroxysmal والconvulsant phases. The catarrhal phase هو هنا يكون microorganism highly contagious. Bordetella pertussis is small, gram-negative, encapsulated, non-motile coccobacilli. There are certain species of the Bordetella which are actually motile. The lovely thing is that when we stain it with the toluidine blue stain, it gives us a bipolar metachromatic granule. That's why يعني أنا ليش ردت هذا الموضوع أشرحه ويا غير بكتيريا اللي تعطينا نفس هم تعطيني bipolar metachromatic granule، بس ما نذكر هسه عم لا تخربطون. So by toluidine blue stain it gives me a bipolar. إذا هنا هاي الصورة أكبرها شوية نقدر نشوف أكو عندي فد bipolar باو هاي كروماتين جرانيولز، تشوفوها bipolar metachromatic granules. هي شو metachromatic granules؟ هي بالحقيقة الميتا هذا الكروماتين يتجمع بالperiphery. Why? Because these are dividing bacilli، فدائما تلقى الكروماتين متجمع بالperiphery عم تنقسم البكتيريا. Since it is a highly replicating bacteria, we see this figure very characteristic to this bacteria. أحد عنده سؤال لحد الآن عن البكتيريا؟ شباب؟ خلي أشكل سماعة. تسمعوني ولدي؟ الو تسمعوني جيد. اعتقد اكو مجموعه ممكن ما يسمعوني. هنا تسمعون ايه اوكي، وهنا همين يسمعوني اوكي كل زين. زين احد عنده سؤال؟ لا اوكي. So the major variants determining of Bordetella pertussis is actually the tracheal colonizing factor. طبعا يساعد بهذا الشيء الفيمبريا والفيلامنتس هيماجلوتينين والبيرتكتين والبارك A. These are actually important for adhesion and colonization on the trachea وعلى الريسبيراتوري سيليا. أول شيء إذا نحتاج البكتيريا هي شنو adhesion و colonization in order to replicate. Then comes the problem where it releases its exotoxin. طبعا عندنا الاكسوتوكسين different types. عندنا pertussis toxin A and B، عندنا الادينيليت سايكليز توكسين، عندنا الديرمونيكروتيك توكسين، عندنا tracheal cytotoxin، وعندنا الاندوتوكسين since it is a gram-negative bacteria, and we have also got the type 3 secretion. طبعا كل واحد منهم له function. الغالبية هي ciliary dysfunction and damage. لو يكون cytotoxic، لو يكون شو اسمه يعني شو اسمه attraction للليمفوسايتس التوكسينز، لكن بشكل عام هو damages the ciliated cells of the trachea inducing paralysis. هذا هو الميجر فانكشن مال هاي البكتيريا. هي بحد ذاتها هي ما تسوي نيومونيا، هي بس تسوي paralysis للسيليا. طبعا how does it overcome the host defenses by host lysosomal inhibition والsiderophore production اللي يسوي host lactoferrin and transferrin are unable to limit iron. إذا فشو تسوي البكتيريا؟ أول شيء it will colonize and this bacteria has got no vector, so it is transmitted directly from one human to another. And there are humans who are actually carriers to this vaccine, and most of the babies get infection from their mothers, which are actually carriers to this bacteria. The organism adheres, multiplies, and it doesn't invade the blood. So there is no specific antibody. You cannot detect the bacteria by a blood test. It liberates its toxin, irritates, and induces cough. طبعا التوكسين they will stimulate histamine اللي هو بحد ذاته همنا يسوي congestion بالmucosa and it actually induces cough. Has a very important effect is that it actually disturbs copurulent streaks of bloody type of discharge. هذا السكريشن will block the bronchioles، فالمايكروورجانيزمز ما تقدر توصل له لللايننج epithelium. It will stick to this mucous secretion and the cilia will push المشكلة اللي تواجهنا. ليش ما طفيته؟ دكتورة الو الو يعني هي تعتبر the best، بس المشكله بيها انه الشلف لايف مالتها كلش قصير. الريجن لو هي افضل ميديا هسه حاليا نعتمدها بالديتكشن مال بورديتيلا بيرتوسيس بالمختبرات. طبعا هاي الميديا كلها بيها antibodies to inhibit the other bacteria from growing. طبعا من طلع عندنا المالتيبلكس بي سي ار حللنا هوايه من اللور ريسبيراتوري tract infections وبورديتيلا pertussis is one of these microorganisms listed in this test. Serology اكو يسوون رابid تستات للاي جي جي والاي جي ام والاي جي اي، بس مشكلتها انه الاميونوجلوبولينز تطلع ورا 3 اسابيع. ف it is negative بس تفيد بحالة الريكفري. نريد نعرف انه بدأت تتكون عنده مناعة او لا او يحتاج لفاكسينيشن. طبعا the drug of choice هو clarithromycin could be replaced by the azithromycin بس لازم ينطي بالcateral stage. بالproximal stage it is of no benefit and بالconvalescent stage همينة is of no benefit. الفاكسينيشن بروجرام طبعا يبدي من الفيرست يير اوف لايف، همينه تريبل توكسويد in combination with طبعا هو part of the DPT vaccine ينطي 3 doses monthly وبعدين ننطي بوستر دوز بالفيرست وال5 years. اكو وايد دول بدها تنطي الفاكسين late in life وبدها تنطي همينه لل early married females على مود يمنع يكونون carrier للفاكسين. طبعا الفاكسينيشن is not protective unless it is taken the precise timing والبوسترز وياها. وكل ما يكون بوسترز اكثر. ليش؟ لان الميموري سيلز لها لايف سبان، ما تبقى لايف لونج. احد عنده سؤال على البرتوسز؟ ارثرومايسين، ينطي ارثرومايسين، اي ينطي ارثرومايسين. بس الازيثرومايسين الحلو به انه هو سنجل دوز، فيعني يعني اعتبرناه شوي احسن واحد من الماكروليدز. الى حد ما شفنا احنا انتي مايكروبيال ريزيستنت لهاي البكتيريا. بعد احد عنده سؤال؟ اوكي، نجي على بكتيريا تسمت على عالم ما اكتشفها، اللي هي ريكيتسيا. ريكيتسيا was actually named after a pathologist. طبعا بالمناسبة كل اللي اكتشفوا هاي البكتيريات هم أطباء، يعني غالبية الباثوجينيك مايكرو اورجانيزم اللي اكتشفوها وبالأحرى كلها هم أطباء، يا إما باثولوجيست لو فيزيشن لو ميليتري دكتورز. يعني نادر ما تسمعون أنه أكو واحد بيولوجيست شخص البكتيريا، غالبيتها شخصوها أطباء. Rickettsia was named after Ricketts, a pathologist who studied tick-borne infections. هذا استمر بالدراسة بالبحث بالtick-borne infections، يعني البكتيريا تنقلها الحشرات اللي تعيش على الإنسان بالطائرة اللي تؤذي تصيب الإنسان. لكن كان عنده فد بروسيجر اللي هو اخذ عليها بوقتها جائزه، وهي كان يمكن سبب من اسباب وفاته الله اعلم، لان هو مات بانفكشن. He used to actually infect himself by the microorganism in order to prove Koch's postulate. فهو ريكتس مات بأحد diseases which were borne by ticks. هو Ricketts described it, but who cultured it and named it is actually Simon Born Woolbridge in 1919. طبعا الريكيتسيا is a very huge family. Some of them are tick-borne, others are non-tick-borne. المشكلة بيها انه احنا كنا دايخين انه هل نعتبرها من ضمن الفيروسز لو من ضمن البكتيريا. ليش؟ لانه it is an intracellular microorganism and it doesn't replicate outside the cell. اكو يعني احنا كل سنه يطلع في الكلاسيفيكيشن للريكيتسيال فاميلي. طبعا they are fastigious bacteria, intracellular, very small, pleomorphic, gram-negative bacilli. With Giemsa stain they appear blue, بس بالميكروسكوب they are very rarely يعني detected و confirmed. الريبليكيشن يصير لو بالسيتوبلازم لو بالنيوكليوس. المجموعه اللي ضمن التايفوس جروب they replicate in the cytoplasm. المجموعه اللي هي ضمن spotted fever they replicate in the nucleus. All members of the family transmitted to human by arthropods except Coxella burnetii اللي هي responsible for the Q fever. طبعا they have a DNA and an RNA and they also have got a lipopolysaccharide. والاكو على السيرفس two antigens اللي هو OMP A and the OMP B, which are responsible for the virulence of this microorganism, and they are also responsible for the adhesion and serology classification. إذا فهي تنقسم إلى 3 groups: spotted fever اللي هي الأكثر واحدة مشهورة بهم ريكيتسيا ريكيتسي، الكونوري والأكاري part of this group. التايفوس جروب عندنا ريكيتسيا باوازاكي والتايفي. عندنا فد جروب كنا فاصلية بعدين رجعناها اللي هي سكراب تايفوس جروب caused by Orientia تسوغوموشي. الثيرد جروب هو transitional group اللي هي هو the most famous one هي Coxella burnetii responsible for the Q beaver, Ehrlichia coffinensis, Anaplasma Phagocytophilomi Ehrlichia wingeli. All these are part of the transitional group. طبعا the disease is caused by arthropod except Coxella burnetii اللي هي is not induced by arthropod. المشكلة بهاي البكتيريا انه it actually is transmitted by the tick. It enters the body and it reaches the blood vessel. When it reaches the blood vessel, it actually causes damage to the endothelial cells, infects the endothelial cells of the blood vessels, damage to it, and induces vasculitis. طبعا it attracts neutrophil lymphocytes surrounding the blood vessel. ومن عندي damage to the endothelial cell, it might rupture and causes thrombosis. طبعا من يصير thrombosis أكثر من part of the body, it causes disseminated intravascular coagulation. من تتجاوز هذا الشيء تقدر it is taken by the macrophage where it actually could live inside the macrophage and it is transmitted to many organs in the body like the brain or the heart. It gives us a very characteristic إذا تشوفوها هنا typhus nodule in these organs. طبعا هنا من يصير vasculitis يصير هذا رش كلش واضح انزين وهنا صار secondary metastasis to many parts of the body. Laboratory diagnosis very characteristic mariola نشوفها بالبلاد film. You can see this blue aggregation of rickettsial microorganisms inside the monocyte اللي هي نسميها mariola. طبعا السامبل مالته هو tissue biopsy من الأورجان CSF إذا كانت واصلة الدماغ and samples taken during the episode of fever. Gilza stain, Reiter stain, the blood film يروينا الماريولا very characteristic. ونسوي immune cytochemistry immune histochemical stains إذا كان عندي suspected tissue looking for the typhus nodule. هذه الكالتشر عندنا هنا modified boarded ganglion agar نستخدمها very useful. طبعا ال requirement 37 to centigrades والresult يطلع لنا between 3 to 7 days. الconfirmation يكون by immunofluorescent plus slide agglutination. Serology همين هنا is of no use because it is يعني يعطيني result ورا 3 weeks of illness. يعني it is not a straightforward detection. Real-time PCR very important, very sensitive. Therapy مالته هو the best therapy هو doxycycline to treat rickettsial infections. نستمر عليه 3 to 4 days. أحيانا بالسevere ill patient chloramphenicol is actually effective. السلفوناميد ما ننطيها لأنه it actually enhances the growth of the bacteria. It is contraindicated in case of rickettsial infections. أحد عنده سؤال ولدي؟